Hi, my name is Tom Minahan, and I'd like to briefly describe to you today about our research program here at Cal State Northridge involving undergraduate and master's level students. Students in our laboratory use the techniques of organic synthesis and molecular spectroscopy, specifically fluorescence spectroscopy, ultraviolet spectroscopy, and circular dichroism, to investigate the binding of small organic molecules to duplex nucleic acids, specifically in our case, DNA. DNA, deoxyribonucleic acid, is one of the most important biomolecules present in living cells. DNA consists of two complementary strands that wind around each other to form a staircase-like structure known as the double helix. Each individual strand consists of a phosphate backbone and deoxyribose sugar with purine and pyrimidine bases attached. The two strands associate with each other by base pairing between the purine adenine and the pyrimidine thymine, or the purine guanine and the pyrimidine cytosine. The base pairs form the steps of the ladder, and the sugar phosphate backbone is the railing of the ladder. The complementary double helical model shown here provides a chemical basis for the storage of genetic information in cells. DNA is contained in the nucleus of our cells and carries information coding for the synthesis of RNA and proteins, the other major biomolecules found in cells. DNA is read in a process called transcription, which gives rise to mRNA, or messenger RNA, which then travels out of the nucleus to ribosomes, containing rRNA, or ribosomal RNA, and proteins, where a process known as translation, which involves tRNA or transfer RNA, converts the information contained in the mRNA transcript and originating in the DNA sequence transcribed into proteins. Proteins are the workhorses of the cell, playing in key roles such as catalysis of chemical reactions and serving as structural materials, cellular gatekeepers, environmental sensors, and so on. However, DNA is at the starting point of all biomolecular synthesis, and thus it controls all chemistry within living cells. The process of transcription, or the reading out of the information stored in DNA, is very complex and involves the participation of numerous proteins known as transcription factors. These proteins assemble on regions of DNA just beyond the coding sequence of a gene, called the promoter, and protein contacts are made at quite distant regions through protein-induced DNA bending. Some DNA sequences, known as enhancers, bind activator proteins which promote gene transcription. Some DNA sequences, known as silencers, bind repressors which inhibit gene transcription. It is therefore clear that protein binding to DNA is a very crucial starting point for all processes that occur in the cell, which are ultimately controlled by the readout of genes. To affect the readout of genes, chemists must be able to craft small molecules which can interfere with these protein-DNA interactions. By binding a small molecule to a particular region of DNA known to be important for transcription factor binding, it is possible either to upregulate or turn on gene expression, or downregulate or turn off gene expression. Most proteins that bind to DNA associate with a portion of the double helix known as the major groove. Proteins often recognize DNA by interacting first with the negatively charged phosphate backbone through positively charged amino acid side chains present on the protein. Sequence specificity is achieved by the interaction of the edges of the purine and pyrimidine bases in the major groove with arginine, asparagine, glutamine, lysine, and threonine protein side chains. Proteins will slide along the phosphate backbone until the correct ensemble of base pairs matching the available protein side chains is found. However, most small organic molecules prefer to bind in the minor groove of DNA due to a better fit and a hydrophobic effect which drives these molecules away from water into the narrow and deep minor groove of DNA. So we need to identify small molecules which can bind the major groove preferentially because some aspect of their molecular shape, steric bulk or size, prevents them from binding in the minor groove. Using the tools of organic synthesis, we're currently engaged in preparing a series of molecules that target the major groove of DNA. These molecules will also possess complementary functional groups which can engage in specific interactions with the edges of the base pairs in the major groove. The challenge is to obtain sequence specificity while maintaining a low enough molecular weight so that these molecules are cell permeable. Once the molecules are prepared, we utilize three forms of molecular spectroscopy to analyze the interaction of our compounds with DNA: ultraviolet, fluorescence, and circular dichroism spectroscopy. These techniques allow us to evaluate not only the tightness of binding of our compounds to DNA, but also give us information about the sequence selectivity when we use DNAs of different sequences, and the location of the binding of our compounds in either the major or minor groove. With the information we gather and with the aid of computer modeling programs, we can then generate a picture of the association of our compounds with duplex DNA for the purpose of evaluating how we can further refine the structure of our molecules to enhance DNA sequence selectivity. Ultimately, we will utilize our spectroscopic tools to evaluate the ability of our compounds to inhibit the interaction of transcription factors with their cognate DNA binding sites. In other words, the small molecule in blue that binds to the same site as the transcription factor in purple has the potential to block the binding of the protein to its DNA target. Regulation of gene expression, an important strategy in the context of current approaches to gene-based disease therapy, may be accomplished with sequence-selective DNA-binding compounds which are capable of promoting or inhibiting the association of transcription factors with their cognate binding sites. Such compounds could serve as important tools for specifically manipulating genetic information processing with a variety of applications in experimental biology, as well as gene-based biotechnology and therapeutics. We would like to thank the many talented undergraduate and graduate students who have participated in this research program over the years, enabling much progress in this area. We are also grateful to the National Science Foundation for funding this research. Thank you for watching this short video about our research program.